Good evening, ladies and gentlemen. My name is Yahya Saif, Muhammad Yahya Saif. I am a postgraduate medical student at University of Warwick. I have been working at the University of Oxford as a VA in dose group, but my research that I'm presenting today originates from Italy in Milan and in collaboration with Aston University based within Birmingham. So the topic of research is investigating the molecular mechanisms involved in early events of cellular dysfunction in Alzheimer's disease. So the project splits into two research areas. The very first one was a mouse model, which gave us the preliminary data, and that was the 5xFAD model. The model was designed in Milan at Mario Negri Institute in Italy, and it was sent over. We performed a lysate of the cortex, and mass spectrometry was performed. Initially, we received 12 samples. The samples were three 3-month-old mice, three 4-month-old mice, three 10-month-old mice, and three wild type mice. So here we're just focusing on the 3 months and the three wild types, because at 3 months the mice were pre-symptomatic and we're interested in the early cellular dysfunction. Once we had the results from mass spectrometry, we used the string database to clusterize the proteins and assign functions. And once we did that, we ran them through a program called FunRich, which gives a biological function to that cluster of proteins. And from the 428 proteins that we had, we thought 128 of them were very interesting in the sense of Alzheimer's disease based on statistical values, quantification, and t-tests. In terms of, there were quite a few clusters, as you can see, almost 25 of them. But from the 25 clusters, we couldn't look at every single protein, so we had to look at specific proteins that we thought were relevant in Alzheimer's disease. The two that we finalized were CRIAB, which is a protein it's a chaperone protein that's involved in regulation of amyloid fibril formation. It's a detangler, basically. And the SLC17A7, also known as VGLUT1, which is a glutamate transporter present in the synaptic neuron. Moving on into the objective of this study. So the previous study that you saw, this is the initial study that was performed in 2020. In 2022, after the initial study, we wanted to validate our results in a human neuroblastoma model, the closest thing that you could get without having to use iPSCs. So the idea was to validate these results, and we wanted to see how do CRIAB and VGLUT1 are affected, or in terms of expression within our human model. So the methods were pretty simple. Culturing was performed. We seeded differentiation with retinoic acid and BDNF. We treated with amyloid beta oligomers, performed immunofluorescence, and finally closed off with Western blotting. It seemed simple, but it wasn't. To kind of begin our investigation, we first had to make sure that the model that we were using was working. Was it relevant to neuronal cells? And to do that, we used the very famous and common method of differentiation, retinoic acid and BDNF. Now, the trick was that when looking at methods available in the literature, you would see that when you treat cells with retinoic acid and then treat them with BDNF for anything less than 5 days, you see cell death, or more than 5 days, you see cell death. Funny enough, when you treat cells with 5 days retinoic acid and 5 days BDNF, they seem to perfectly show a neuronal-like morphology. And I think the flaw that a lot of methodologies have is when they're treating cells with BDNF, they also leave serum in the media, which is a heavy problem because BDNF and the serum compete with each other in terms of differentiation. So we used serum-free media. Now, we added, as I said, soluble amyloid beta directly onto the cell. And as you can see in the control type, you don't actually see any fluorescence suggesting that there is close to nothing of amyloid beta being detected. But when we do it for the cells that are treated with 1 micromolar A beta for 72 hours, so they get 2 treatments, one after every 48 hours. So they get initial treatment, then a change in treatment after 48 hours. And you can clearly see the increase of A beta detected within the cell. Now, to get a better understanding of what it actually looked like in maximum intensity projection, as you can see, no A beta here, some indigenous, but it's negligible when we quantified. And when you look at the treated cells, you can see clear fluorescence of increased amyloid beta within the cell. So, out of interest, we thought we've treated the cells with amyloid beta, which is not a very common thing to do. Normally, the treatment involves transfection, but we just treated cells extracellularly with amyloid beta. Now, when we did that, we thought, does it actually have an effect on the viability of the cells? So we looked for apoptotic nuclei under the Leica confocal microscope. And funny enough, yes, we quantified 7 different experiments. So 5 biological replicates and 7 technical replicates. I think the PowerPoint is of it hasn't updated. So this is supposed to be a super plot. You can't see the rest of the data and you can't see the significant values, but the p-value is lower than 0.0001. And as you can see pycnotic nuclei, and we screened the entire well to get a representative understanding. Next, one thing that we didn't think we would find in our cells, because you can do a PubMed search on this, nobody's ever looked at this before, and what we call it is neurite fragmentation. As you can see, when we treat cells with A beta, you see that the neurites become fragmented. Now, the interesting thing is, those neurites, the neurite is fragmented, but it's still attached. If it was a clear sweep through from the neurite one pin from the one pin to the other pin, you would assume that the rest of the neurite would disappear. But the fragmentation is there in the neurite, but the neurite is still intact. So we can assume that only one side of the cell is fragmented and not the other, hence why it's keeping intact. This obviously isn't something that's been explored before, and it's not available in the literature. So this was one of the key findings in our validation. This might be just relevant to this model of SHY models, but who is to say? We need to find out. Further investigations need to happen. So the trouble and the nightmare of fragmentation is quantifying. How do you quantify an image that shows fragmentation of the neurites? ImageJ is a beautiful program, but it's not good enough. So what we did is we designed a program. And when I say program, we wrote a code that helps that calculates degeneration index. And what it does, it takes sizes of the neurites and it looks at fragments. Every time that neurite breaks, it will consider that a fragment. And we compared the fragments within control cells and we compared them in the A beta treated cells. As you can see, the first was performed in EVOS 500, and the second was performed under Leica confocal. The EVOS 500 shows significant fragmentation, and the Leica confocal shows a increase in fragmentation in both of the replicates. When you compare each experiment, they were significant. But when you calculate a mean for both experiments, it's not a significant result, but there is definitely an upward trend. So this was a bit difficult to kind of get around. Now, going back to the initial idea of validating the proteins that we thought were interesting, CRIAB initially, the finding happily matched what we saw in the mouse model, the 5xFAD mouse model. And you can see in immunofluorescence, there is in A beta compared to the control, there is a reduction in immunofluorescence. And that reduction for each experiment was significant. Again, when you calculate the mean, the significance is lost. But every biological and technical replicate that we performed, the result was significant. And we thought that we had an idea that yes, CRIAB is decreased, but this also required further investigation, which I will touch up later in the presentation. Now, the second protein that we thought was also interesting was VGLUT1. Now, VGLUT1 has a lot of mention in Alzheimer's disease if you do a simple PubMed search. And when we looked at VGLUT, there was a clear increase in immunofluorescence in the A beta treated cells compared to the control cells. And as you can see, that the result for all 3 biological and 9 technical replicates was significant, and even the overall value was significant. The p-value was less than 0.05. So immunofluorescence tells you a lot, but not enough. So we decided to turn to Western blotting to quantify the protein. Now, biology is nice until it's not. And we saw the nasty side of it when we did the Western blotting. When we performed the Western blot, the control cells had, they had less CRIAB compared to the A beta. Now, the moment we found the results, we were like, okay, this does not make sense. And after doing a bit of reading up in the literature and how CRIAB is affected within the cell, within the pathology, we very quickly found out that the CRIAB that we're seeing there could be phosphorylated version of CRIAB. Because the samples in a Western blot are lysed, it is very, very likely that the CRIAB that we have that is increased is the lysed with the phosphorylated version of the CRIAB. And we know through multiple studies that P38 MAPK causes a lot of phosphorylation of the CRIAB. Obviously, we need to validate this, and we're currently working on this to confirm that this is phosphorylated version of CRIAB that is showing the increase, and not the functional CRIAB. In terms of understanding why CRIAB is important, I did say that CRIAB is a chaperone, it's a detangler. So in normal, in a healthy control, you would see that you have A beta plaques. A beta plaques are something that happen all the time. It's happening in our head right now, but it's happening in controlled quantities. And our body is able to respond to that and detangle those deposits. So CRIAB intervenes and it detangles the amyloid deposits. But when it comes to AD, there's 2 things. The deposits are increasing, so there's higher frequency and higher load of A beta deposits and plaques, and the function of CRIAB, the detangler, is also reduced. So you're having a double, like we call it in the UK, a double whammy situation, and it increases and it boosts, turbo boost to the pathogenesis, and you see a very detrimental response in terms of the pathogenesis increasing quite significantly. Now, going to the Western blotting that was performed for the VGLUT, this time the results matched up the immunofluorescence, and we can clearly see that VGLUT1 is increased in cells that were treated with A beta. Now, the trick is VGLUT1 does increase pre-symptomatically, but through the data, as I told you, we did have mice that were 4 months and 10 months, and we see as you go along that VGLUT does come down, and there's a reason for that. We believe that the initial influx of or the excitation of glutamatergic synapses is a response to the loss of synapses that occurs in Alzheimer's. So because you're losing synapses, the body tries to use whatever synapses are remaining and intensifies the usage by stimulating them more, and you get more glutamate being fluxed out. And what that does after a little while is it causes excitotoxicity. So you're overusing the synapses, the glutamatergic synapses, and even the other synapses. And after a little while, those synapses aren't able to cope again, and you're losing those synapses as well. So not only are you losing synapses because of plaque deposits, but you're also losing synapses because you're overrunning the synapses. And there is a really nice article on excitotoxicity mentioned right here, which goes further into how the molecular mechanism works. So to finish off, acknowledgements of the people involved, Dr. Marilina Rapici, one of the best people I've ever worked with, and some of this research, it belongs to the Mario Negri Institute, and the quantification was helped by the people in the last lab that I was working at University of Oxford. And you have my contact details if you wish to know more or would like to collaborate on something. But thank you very much. This project needs a lot of work, and we're looking at data that is very promising, and it requires a lot of funding and a lot of manpower. So hopefully in the next couple of months, we should be able to present something that might change the outlook on the pathogenesis. Thank you.